to Dr. Glynis' Chicken Genetics videos. It's a common belief that you can tell what colour eggs a young chicken might lay by the colour of her earlobes. In this video, we have a look to see how true this might be. I'm sure we all know our chicken's ears are hidden by fine protective feathers, and maybe you also realise that the earlobes are often clearly visible. Earlobes are flaps of skin under the ear canal, present in both hens and cockerels, and they tend to grow larger with age. They come in a variety of colours. They seem to be white in red jungle fowl, but in domestic chickens they may also be red, pink, blue or green. For most commercial egg-laying breeds, the colour of the earlobe is associated with the colour of the egg, so that we can predict egg colour from the earlobe colour of young hens before they start laying. Those hens who will lay white eggs have white earlobes, and those who will lay brown eggs have red earlobes. For tinted egg layers, the earlobes are pinkish or red with a white spot. Our head of biology, Professor John Fish, used to tell the new students every year that for every rule in biology, there's an exception. Professor Fish was, aptly enough, a marine biologist, but I used to think his saying applied well to genetics too. And here we certainly have exceptions to the eggs and ears rule. Interest in earlobe colour dates back to at least the 1920s. The famous chicken geneticist Dr. Warren published the first scientific paper about it in 1928. More recently, there's been a couple of molecular genetics studies shedding a little more light on it. I've left references below for anyone who wants to look. Dr. Warren discovered that the genetics of earlobe colour is polygenic and complex, being affected by many genes on both autosomal chromosomes and the Z chromosome. He also found that breeds having the same earlobe colour could differ considerably in the genetics of this character, which again indicates several genes could be at work here. On the other hand, individuals within a single breed or strain were similar, but differed in some genes with relatively minor effects on the phenotype. Together, these show how selection has worked within breeds with enough variation for there to be significant differences between breeds. Significantly for this video, Dr. Warren also found, and I quote, "no evidence of any linkage between the factors for earlobe colour and egg colour." Dr. Warren published this data almost a century ago now, at a time when there were no computers to analyse data or draw pretty graphics. So I have taken this data on eggs and ears and plotted it for us using a spreadsheet. Dr. Warren graded the egg colour from 1 to 9, with 1 being white eggs and 9 being dark brown eggs. He also categorised the earlobe colour being either white, red, or predominantly white and predominantly red. I have plotted the frequency of hens laying different coloured eggs, with different bar colours representing different colours of earlobe. It can be seen that there is no evidence for any association of white earlobes with white eggs or red earlobes with brown eggs. Hens with different earlobe colours lay eggs of almost the same average colour. Dr. Warren concluded that the existing association among some breeds was probably due to the origin of the breeds. Selection for breed standards, for example, could have produced an apparent association which can break down without crossing. More recently, there have been a couple of molecular genetic studies that have identified regions of the genome, including possible genes associated with earlobe colour. One of those genes looks as though it could be similar to a gene involved in blue egg colour. It seems to me that both studies indicate that earlobe colour is polygenic, with one or some genes being sex-linked and at least 2 being autosomal on chromosome 2. I will put the references below. Perhaps in the future, further molecular studies will increase our understanding of earlobe and eggshell genetics, but for now we can conclude though the association between the two most likely comes about because of our selection criteria during breed development, perhaps with an element of chance. The association isn't an absolute one and breaks down when we start to take a closer look. This is something that has been known since Dr. Warren's day, but we haven't always been aware of that. Thank you very much for watching this video. If you have found it useful, please like, comment below, and share it with your friends or on social media. Subscribe so that you're notified of chicken genetics videos as they come out. I hope to see you very soon.